So in differential gene expression studies, we come up with hundreds of genes which are differentially expressed. So since we have large number of genes, it's hard to find what biology is going on. So they have provided us with what we call them as gene ontology annotations or Go terms. And there are three different classes for these Go terms are there. These Go terms or gene ontology terms are implemented as directed acyclic graphs. Remember guys, people try to find out hierarchy in those Go terms, but actually it's not there. They are not trees, they are kind of graphs. So there we have the terms which are related with genes, many genes, and one gene might be associated with many functions or many terms. So in this way we do have a many-to-many relationships, and that's why they are implemented as DAGs or directed acyclic graphs. So there are three main categories or classes of these Go terms. One of them is called as biological process, or B represented as BP. We have cellular components, CCs, we have molecular functions. So you can look into what cellular components are there or what biological processes are under control of these list of genes. Mostly people, they report those molecular functions in their research papers. So here is how it looks like. So for example, at the top we have a very generic term, and then these terms, they keep on breaking down into the specific ones. We see that there may be the terms which are kind of associated with one another, and then people try to capture the hierarchies amongst them. But since these terms are associated with many genes, and at the same time one gene may be associated with many terms, it's hard to define true hierarchies among these Go terms. DAVID is an interesting tool. DAVID stands for Database for Annotation, Visualization and Integrated Discovery by Huang et al. You can find it on its web link, which is given down there. So most of the people in these differential gene expression studies, they've been using this DAVID. It provides a comprehensive set of functional annotation tools to understand the biological meanings behind the large list of genes. This is the page for DAVID. If you go into it, you can go and click it into functional annotation link. So once we go there, you can there is one thing called as upload. You can go up and press this and you can upload your list, and you can attach a file with these. You can check if that file contains a multiple list, or you can put your gene IDs into these text box. When you click your Go button, you need to also recognize what kind of IDs or gene IDs are there. If it is you don't check anything, then DAVID tries to calculate what gene IDs are provided. So here we have ATs, which are Arabidopsis thaliana gene IDs. So that's why I checked here Arabidopsis database actually, and these are those IDs which are coming from that Arabidopsis database. There is also two radio buttons. You can check whether it's a gene list or it's a background. So sometimes you come up with those your gene lists and you want to test against another list as the background. So you are trying to look into the enrichment of those functions in those 100 genes, for example. So for example, you have 100 genes, and among those genes, your one of the functions, like for example your reduction oxidation, is present in say 20 out of these 100 genes. So you can check it against the background. So that background can be your whole individual, whole human being, or whole Arabidopsis, all genes. So what is the proportion of this particular function in those all genes? So while it compare their relative proportions, it comes up with an enrichment score or a p-value. So it recognizes this list. Here are different options where you can check it. So you can check its functional categories, you can check the gene ontologies, look into general annotations, literature related to it, and pathways, and etc. etc. There is also a tool called as functional annotation clustering. So you can get these Go terms and cluster them together based upon how many common genes are present between these terms. You can get their charts, you can get their tables. So here is one example. It's called as BPFAT. So BP stands for biological process. FAT is where it tries to take all those terms and try to put the generic terms and specific terms all together. So in this way, it's kind of, most of the earlier tools, they were using a method called as SLIM, so in which they were trying to converge the terms together. So from specific to generic, they were trying to merge those terms. Since we get a large number of Go terms, so it's hard to capture the big function or the big biology going in it. So they were trying to do those converging them together. They call them as SLIM. So in order to stay opposite to that, they would use the word FAT. So here we see those terms. We have their number of genes in those terms, and then we come up with their p-values, and then we have their correction. Since we are doing multiple tests together, so we need to take these corrected p-values. So here this correction method used is called as Benjamini. I'm not going into the detail of it, but it's a multiple test correction method. For cellular components, for the same list of gene, we see that there was around 954 genes which were present in this. We came up with 15 terms. So remember, there are many genes associated with these terms, and then obviously these list of genes, they have intersections. Same way we can have molecular function terms. So we have, for example, binding, ion binding, cation binding. So these terms are together in this category. David also tells you which pathways are affected as far as these genes are concerned. So these genes are controlling which pathways. You can understand the biology while looking into these pathways. There is another cool tool, we call it as functional annotation clustering. So that is, it takes all those Go terms and bring them together based upon how many common genes they have. That is, it goes with their intersections and make up a clusters for them. So in this way, they are arranged into these clusters. So top one, cluster 1, and that is has a score. So the more genes are common between the terms, higher is the score. Then comes cluster 2 and so on. So in this way, within those clusters, you can look into very generic terms and very specific terms also. So in this way, you can derive your biology, what biological processes are going on. We can see the terms coming from molecular functions, cellular components, all of them, they are merged together over here in these clusters. So you can have a better idea about what biological process is under control, what functions are there, what pathways are affected while looking into these clusters. We can take these clusters and then we can plot them together into this pie chart where we can have a relative proportion of what functions are, for example, taking the bulk of these genes. So for example, serine, threonine kinase are present as a bigger proportion as far as these Go terms are concerned. And same way, there are other categories also. So Go term analysis is an interesting process where we can take the list of genes. Since we have hundreds of the genes, we cannot present those genes individually. Some of the genes in which we are interested, we can present them individually, but as a whole, in order to infer a generalized biological pattern, we need to do this Go term analysis.